Hi, I'm Liam Macaulay and welcome to my presentation discussing the hormone ghrelin. Specifically, I will be discussing ghrelin's cytoproduction, regulation, target tissues, physiological action, cell signaling pathways, and disease caused by altered function. Ghrelin is a peptide hormone and is produced by specialized cells found in the mucosal layer of the gastrointestinal tract. Although these cells can be found throughout the gastrointestinal tract, they are mainly concentrated in the gastric mucosal layers. If you take a look at the image presented on this slide, you can see the gastric layers of the stomach. The mucosal layer is divided into three layers: the epithelium, lamina propria, and muscularis mucosa. It is the epithelium that contains the ghrelin cells. Ghrelin, also referred to as the hunger hormone, plays a vital role in energy homeostasis by stimulating an increased desire for food consumption when fasted. So it should come to no surprise that during a fasted state, plasma ghrelin levels are increased. Not only does fasting regulate the level of ghrelin found in the plasma, but so does intake of nutrients. Plasma levels of ghrelin are found to return to basal levels after the consumption of a meal, showing an inverse relationship of food consumption to plasma ghrelin levels. These changes in plasma ghrelin levels are quite visible in the diagram to the right, which shows the rise in plasma ghrelin levels prior to breakfast, lunch, and dinner, and the rapid decline post-food consumption. Alongside fasting and nutrient consumption affecting plasma ghrelin levels, several studies have suggested many hormones play a role in the regulation of ghrelin. For instance, somatostatin, produced by D cells, plays a role in the inhibition of ghrelin secretion from the mucosa, and glucagon has been found to stimulate ghrelin secretion. Not only is ghrelin affected by other hormones, but ghrelin itself interacts with many other hormonal systems. An example of this is ghrelin's role in triggering the growth hormone IGF-1 axis to increase muscle hypertrophy. When ghrelin is secreted from endocrine cells found predominantly in the gastrointestinal tract, but also potentially the brain, keep in mind there is somewhat of a debate about whether ghrelin is secreted by specific cells of the brain. It travels in the circulation, where it moves towards target tissues, where it is involved in producing a state of hunger, responding to stress, learning, memory, and motivation. Ghrelin has this ability to influence many physiological processes by binding to a receptor. The image on the left shows this interaction. The receptor for ghrelin is the growth hormone secretagogue receptor 1A. This receptor is found in many regions of the brain, such as the hippocampus, thalamus, cortex, pituitary gland, and hypothalamus, as well as in some peripheral tissues. Although the growth hormone secretagogue receptor 1A is found in many tissues of the brain, it is mainly concentrated in the hypothalamus and pituitary. The growth hormone secretagogue receptor 1A is a G protein coupled receptor that, when bound to ghrelin, interacts with G proteins to activate them and begin intracellular communication by a signaling cascade aimed at amplifying the signal. The cell signaling pathway generally produced when ghrelin binds to growth hormone secretagogue receptor 1A involves the amplifier enzyme phospholipase C, seen at the top of the diagram. Phospholipase C is able to produce two different second messenger molecules, IP3 and DAG, seen to the right of phospholipase C in the diagram, by breaking down membrane lipids. IP3 goes on to bind with receptors found on the endoplasmic reticulum organelle, as seen in the bottom left of the diagram, which helps to release calcium and increase intracellular calcium levels. DAG also plays a role in increasing intracellular calcium levels, but does so in a different way. DAG activates protein kinase C, which is able to block potassium channels, which causes the membrane potential of the cell to become less negative, leading to the opening of voltage-sensitive calcium channels and an influx of calcium into the cell. An increase in intracellular calcium, a second messenger itself, can lead to all kinds of physiological effects, such as growth hormone release in pituitary cells, the release of neurotransmitters and hormones, and changes in gene expression, as well as many others. Considering ghrelin is required for so many physiological actions in our body, it is essential that ghrelin's function itself is not altered, as it can play a role in diseases such as anorexia nervosa. Anorexia nervosa is a disease where people are severely undernourished and underweight, as seen to the right. People who are suffering from anorexia nervosa, though, surprisingly show high levels of plasma ghrelin, which is quite a paradox considering that ghrelin stimulates hunger, making people much more likely to consume food. So the question begs, why do people with anorexia nervosa have such a difficult time gaining weight? even with such high levels of ghrelin. This is likely because of genetic variations and because of impaired ghrelin signaling. The exact effect of genetic variations are unknown, but impaired signaling means that calcium influx into the cell, as mentioned previously, would be affected, resulting in the impairment of growth hormone release, the release of neurotransmitter and hormones, and gene expression, which all could play roles in stimulating hunger. That is all for my presentation. I hope you learned a thing or two. Thank you.